In BioClonal, we are very focused on a biomolecule called an antibody. It's a complex biomolecule because it's very big and it can be used for different things. With our platform, we can build like super immunogens and catalyze the mechanism so we can obtain the monoclonal. It all began with curiosity. I was this kid always asking why, trying to figure out how everything works, trying to learn how life works in a molecular base. My aim from that point was to bridge between basic science and the market that at the end goes to the patient.
What if the biggest challenge in cancer detection isn't the cancer, it's the engineering behind it? Jessika Valero Gonzalez, Chief Research Officer at BioClonal, is working on a problem most people don't see. Many promising cancer proteins are nearly impossible to detect because existing animal models fail to generate the right antibodies. Jessica's team uses inverse engineering to create immunogens that unlock antibodies for proteins other methods miss. In this episode, we talk about how rethinking the engineering process is opening new pathways in early cancer detection. Let's get into it. Welcome to the show, Stories in Life Sciences. Excited to have you on here today and hear more about your journey with BioClonal and then just leading up to the point. Where are you calling in from today?
I'm from Spain and I'm based here in a city called Zaragoza, which is in the north of Spain.
Today, you just kind of want to start with that for us, like how you led and your past and your history with biotech and how you got to where you are today.
It all began with curiosity. I think from the very beginning, I was this kid always asking why about things and trying to figure out how everything works. So I think maybe because of that, I choose to study chemistry here, also in Zaragoza, trying to learn how life works in this molecular base. Once I finished my bachelor in chemistry, I moved to biochemistry. So I did my PhD on protein structure and enzymatic properties of the different proteins. And once in that moment, I realized that I really liked research, but I couldn't see how this basic research, finished in patients. So I decided to let the academic research in that point and start working in the private company. My aim from that point was to bring this or do this bridge between basic science and the market that at the end goes to the patients.
Perfect. Yeah, I see a common thread there with the academia into out into the business aspect of it. It's like you really want to see those projects that you're working on actually get out there into patients and want to see part of that profile. So then, where did you start at when you left academia?
I started working in Certes Biotech. Now it's a big company in the diagnostic field. When I started working in there, it was not so big, so it was also a very nice and really rich time for me to learn how a company could grow and how to do it, let's say. So I started working as a researcher in Certes Biotech, but soon I realized that I like to see where these products or where these developments were. So I like to go to the hospitals. I like to go to the final clients to see our products working in real life. So I was for several years there, and at the end I was a business director unit of one of our technologies, and that was very nice and helpful for my career to learn how to build a whole business unit and bring this science to the patients.
What were some learnings I get to transitioning from academia into the private sector? Like some maybe some challenges too that you experienced, like going from there to the academia to that world.
Oh yeah, there were so many challenges. The academia world and the private world is totally different. Both are very competitive. but in a different way. When you are in the academia, you have a lot of competitors, including your own partners in the lab. And the first competitor is yourself. You compete with yourself every day to see how far you can go, how fast, and better every day. Because the publishing is always in your back, because the money in the group depends of that. So you are fighting against yourself all the time. And of course, in the private company, it's also very competitive. What I feel in the private company is the team building is absolutely important, because you want to go farther, no faster. So you need to build this team from the very basis, and so you are able to compete with your stakeholders in a proper way. So I think it's different, and to learn how to change that chip, it's difficult.
Yeah, I didn't think about it in that way. Just like the individualized aspect of it compared to the team part of it. I mean, how did you learn that? Like, did you have any mentors or people that helped you, or?
Well, at the beginning, no. I mean, I started working. I think now 13 or 14 years ago in the biotech sector, and in that time, at least for me, was not so many mentors around. Now there are so many coaches, and it's more in the
Yeah,
move. But at that time, no. But I found it. I mean, I got very nice responsible for my areas. I learned a lot about my general manager. I have a very good scientific advisors also inside the company. So even though we didn't have that specific names or these specific roles back then, you find out who can help you in what, if you do the correct questions and in the correct time, you can find it.
I agree with you. It's interesting. There's so many of these incubators
Yeah.
and different places now where I feel like some of these founders can go. They have all these
Mm.
mentors, all these opportunities to. But if you just went straight, like I did feel like did too at a young age into the private sector, a long time ago, you just didn't have that. You had to find
Mhm.
it, but it does exist.
Mhm.
I feel like so. Yeah, good for you that you found that because I don't feel like it was always easy to find.
No, it's not easy. And here in Spain, we got that point that we are like very friendly people, but we don't like to share
Mm.
so much. So yeah, it's like that. So you have to find the proper person and the proper moment, and of course, be able also you to share. You have to be open also.
Tell us a little bit more then about that transition to BioClonal. Like why did you make that transition, and then a little bit more about what you're doing there, what your role is.
Mhm. Well, I moved to BioClonal 3 years ago. It's a totally different company and totally different purpose. And I joined it because I like to build projects. So once
Mm.
I've been in Thortes for several years, I found myself like I wanted to build something new. So I joined this team. It was a very small team at that time. We are growing slowly because we are a startup. And we are building a biotech company based in therapeutics. So different field, different stakeholders, different size of company. So everything new. But I really like that feeling of that I'm building.
How did you find it?
Well, we are related somehow to Thirtes because one of the owners of Thirtes is also the owner of this new company. So we are like
Uh,
a spin-off, something like that.
can you tell me more, just the broader scientific vision and commercialization? Just dig a little bit more into BioClonal
Mhm.
as far as what you're focused on, and then where you're at in that.
Yeah, well, in BioClonal, we are very focused on a biomolecule, which is called an antibody. Of course, you all will know something about that by that biomolecule. It was very we got all in the news in the COVID about if we got or
Yeah.
not these antibodies of the if we got the infection and our antibodies growth. That's the biomolecule. It's a complex biomolecule because it's very big, and it can be used for different things. It can be used for diagnostics. For example, this typical test for the COVID when we use the swab and we get like two red lines if we get infected.
Mhm.
That are there are monoclonals there, antibodies in the red lines.
Really?
Yeah,
That's even in the testing.
yeah, yeah, yeah,
Okay. That's more broad. I
yeah.
didn't realize
So
that.
if
Okay.
you can imagine that test, and you can imagine this invisible line, there were monoclonals. They are like Y shape in that position, waiting for your sample to go through them. And if the sample
Oh.
gets the infection, if it's infected, and there are proteins of the virus there, the monoclonal catch it and appears the color in the line. That's the more or less the technology behind this rapid test. So that's one of the applications for monoclonals, and that's where I learned from antibodies in my time in diagnostic. We use monoclonals for our test. We design our antibodies for the specific test for the specific antigens. And well, the other application for these antibodies is to use it as therapeutics. Imagine that you have this biomolecule and you put it inside your body. This antibody can bring some can attach for some antigen that is in your body, or can attach a specific cell, for example a cancer cell, and can tell us where it is. So they are very very specific as markers and also as treatment tools. So what we do in BioClonal is to use all our knowledge about this complex structure into the therapeutics field. And how we do that? Well, we have a specific platform that enable us to obtain these specific antibodies for challenging targets. This challenging target word is like very broad concept, but inside of that concept are all of those proteins or things that in your body are difficult to express. So, for example, let's give you an example. When we are talking about cancer cells, they are different. Every
Okay.
cancer cell is different because it has a different antigens or different things put inside the surface. Um, with our technology, we are able to obtain monoclonals against specific cells or specific antigens. But with another technologies, this is not so easy, or it's not even possible. So with our technology, we obtain this monoclonal that can be used for treatments or markers for different diseases.
Wow. So for specific cells,
Well,
like that
yeah,
specific.
it could be so specific if the antigen is specific. It could be so specific. Yeah.
Wow. And why is it that you think I noticed that these monoclonals have been used for different therapeutics in the past? It seems like not so much for some of these other disease states that you all are focusing on. I'm just curious as to why. I know
Hmm.
when I look at it, it's more difficult, it sounds like, which is obviously probably the main reason,
Mhm.
but like, why is that more difficult? And then why, I guess with BioClonal, why are you all focusing on that, which seems to be more
Hmm.
challenging?
Yeah, it's challenging. Actually, we just have like 140, 150 monoclonal antibodies approved by FDA today. There's not so many. It's a
Yeah.
quite difficult molecule because it's very big, and they have to have like several characteristics as many other drugs, of course, but as this is so complex, it's difficult to check all the boxes in this list. And one of the things that is the reason why is quite difficult to have these monoclonals is the reason why BioClonal is here. Till now, because we can talk about later about what is going to happen with biotech and AI and machine learning and so on. But till now, the monoclonal development is based in animal models, basically in murine models, rabbits, and chickens, but mainly murine models. Why? Because the same reaction that we are looking for in the human body when we get the monoclonal and the antigen, we use the same mechanism in the murine model. So the mice or rats, they give us the monoclonal. So we do like inverse engineering. Okay, that's the common way to obtain the monoclonal. We select, or well, the technician say, okay, this kind of antigen is, or this kind of protein, it will be useful to detect this specific type of cancer. Okay, so you take this protein, you inject that protein inside a rat, for example. So the rat generate a monoclonal. It's a simple way, but let's resume like that.
Yeah.
So you
Simple's good. Simple's good.
yeah. So you get the monoclonal and you study all the things that this complex biomolecule needs to have to be used as treatment in a human. So once the human got this protein in the blood because of the cancer, this monoclonal will catch it. Okay, that would be the circle. So this is very resumed, but in the practice, it's quite long and quite difficult. Why? Because one of the main reasons is, what about if this protein doesn't cause any response in the rat? So I don't get the monoclonal, so I cannot use it. Okay? And why this rat should not cause a response? Well, there are so many proteins that are very similar between humans and rats and or chickens or different animals. They are very, very similar. Such similar that sometimes they find it like they are their own proteins. So let's say that nothing happens. You use this protein in the rat or in the mouse, and they are peacefully lying. So the process doesn't start. With our platform, what we do is this kind of proteins or antigens, we can build like what we call super immunogens. So the rat or the mice found it strange and catalyze the mechanism so we can obtain the monoclonal.
So to lessen that failure rate, that obstacle.
Yes, that's it. We break that barrier. We let the mechanism work.
As much as I was reading on it too, that makes more sense. So that makes sense as to why you're an open platform essentially on that part of the process. So you could work with other drug companies or development, because that part of that, which has been challenging before, where they could have something that would fail, with a mouse, you've developed a way to where you've limited or eliminated that process.
Exactly. So we can work for third parties, or we can collaborate with them in a specific projects that this kind of approach could add some value. And we are on it. We are opening our platform for third parties, and we are also working in our own developments. We want to have a very short pipeline, because you will already know that the biotech sector, it's quite expensive. every part of the pipeline is expensive, but we have our own pipeline based in Alzheimer's disease.
Yeah, this seems creative too, in terms of, like you said, it's so expensive. So at the same time that you're working and developing your own, you can have a different revenue stream as to opening up the platform, so you can still develop that while you're funding and doing your own larger projects. Is that kind of the
that's the idea? Yeah.
From an overall perspective, getting creative, almost like creative funding ways, essentially, without just taking on all this investment from the outside. So when you get to the end, it's so diluted that you're finding ways along the way to use your technology for other things while still developing your own. Yeah, that's exciting to see.
Yeah, that's it. That's the strategy. I'm also feel like it's also very interesting for this kind of companies to be in the market soon. That is something that I learned in the past, because well, it's quite easy to listen, but it's quite difficult to really understand what the market needs. And sometimes when these developments are so long in time, you can spend like 5 years under a development, and once you finish, you realize that no one's wanted. So
so sad to me, but it's funny. It's not funny. Yeah, it's true though.
Yeah. So for me, it's very interesting. Of course, the revenues are interesting for everyone, but it's also because we can learn faster if we are in the market.
No, that makes so much sense to me to do it that way. I feel like in the past, I remember that when I was in my like biodesign program, you see so many, they worked on something in a lab for 5 or 10 years, and by the time they got it out, the market didn't really need it anymore. That's a lot of time and money. So this, to me, it's innovation can happen so much quicker, and we can actually come out with things in the end that become real products and can help.
And nowadays,
goal,
and nowadays it's even faster. I mean, I have the feeling that everything is running around me so fast that you cannot plan in 2 or 3 years how is going to be the sector or how is going to be this solution. It's quite difficult. So if you are not there in the market, listening and really understanding what they are trying to tell you, I think we are going to be quite lost.
Yeah, I agreed. Plus, this seems to me is a better funding strategy too. Not just relying on so much intensive capital up front for these long lengths of time that you can actually bring more things to market, with this way too, and bring more things quickly to market.
Yeah.
So that's what it feels like to me.
Yeah, that's the idea.
How did the initial discovery happen? Like BioClonal, like the discovery that, okay, this, first of all, how it got discovered, and then, we should make this a business, like its own business, a spin-off.
Well, I think this is like a relationship. It's not one day of the beginning, yeah, it happens like very, very natural. The technology was, well, one of the owner of the company, one of my colleagues, which is the chief scientific officer, has more than 40 years of experience in monoclonal antibodies. So, yeah, the idea was building step by step for several years. And for us, well, for the holding I belong, the COVID situation was quite wealthy in terms of the financial. So they decide to reinvest in biotech, which is I think it's quite challenging and quite well, to be honored about that. So when this idea was already there and the moment was there, so I decided to put these two pieces together and start the project.
Okay, so timing, a lot of history, a lot of science that went into it, and then just the market timing of it as well. So, okay, that's where it came from. And I noticed Alzheimer's is a specific disease or therapeutic state that you all looking at, which is exciting. Obviously, that's a big market. Can you tell a little bit more about that, and I guess why the focus and what that does, what your technology specifically, how that treats it?
Yeah. Well, the reason why we choose Alzheimer actually is a biochemical question. we all in the team, we try to understand everything from the very basis, from the molecular basis. That's our focus and our move in the development in our research. So the thing is, we found a specific target, which is already published, which is a very strange molecule which is related or is supposed to be related to Alzheimer. So we decided to try this small protein with this complex wave to prove that our technology works. In the past, a company tried to obtain monoclonals for treatment in Alzheimer with this target, and they failed. So we decide that even though it's challenging, it would be very interesting to use this structure to prove that our technology works. And in other hand, we are quite familiar with Alzheimer, which is not so like again like two things happen at the same time. And say, okay, if our technology could work for this, and we want something for this, let's put it together and start working in this molecule. We don't know how far we are going to get. We are getting the first candidates against this molecule, but once we get the candidate, the way to get the drug is so long and so expensive that we don't know exactly where we are going to stop. So our first objective is to prove that the candidates are working with this protein, and to prove that the protein is in the patients. Once we get there, we will see which is the next step. We don't know.
Got it. Yeah. You say before, like everything's moving so fast. That part is the part that has not started to move fast. Like actually getting designated patients, doing a study, testing that part. It just seems, yeah. I'm excited about maybe new ways to do that as well, to test things, to get things out there without that. I mean, you mentioned like AI a little bit earlier. I'm just curious, like how you see the potential for that maybe within biotech, or what you see. I don't know if it relates specifically to what you all are doing, but what you see the influence of that may have, within your sector.
I had a class today about AI, and every step I took in the AI, I feel like I'm even more lost. It's For me, it's very difficult to find how AI is going to help biotech. I'm sure it's going to help it, but for me, it's very difficult to find it. For example, there are so many tools, quite good ones, from the beginning of the developments, where you don't know, for example, the structure of something, and you get just with the sequence, you get the structure. There's a funny story about it. My PhD was about crystallography, which is a technology that enables you to see exactly like a photo of the protein structure. So it's been used for several years to learn how the proteins work, or the enzymatic, or the DNA. The alpha helix of the DNA was saw for the first time in crystallography. So I finished my PhD with several structures, which takes me like 5 years to obtain. And just 2 years after my PhD, or even sooner, the AlphaFold comes to life, which is a tool that with the sequence of the protein, they give you the structure in seconds. So that was that in that moment I realized that something big is going to happen here.
Yeah, no, I'm excited for I was at a conference. So one of the leaders for Google with AI talked about that specifically, how what used to take years of one person focused on just one, that now they solved for all.
Yeah, that happens to me personally. So I can see specific dots that the AI is already helping us. And for example, in the patients trials, I think there is going to be very, very helpful. There are already some companies working with twins that could help us to understand how the drug is going to work in the metabolics of the patient. So, of course, it's not the whole trial. You cannot do the whole trial with that tool, but maybe we can reduce the time of the phase 1 or phase 2. So there are things that going on, but there are so many big block spots that I cannot imagine
Yeah.
how is going to be there, the AI. But I'm sure we will figure out soon.
It is interesting to see. I know when you start dipping into that world more, sometimes it just becomes, it can be overwhelming,
Yeah.
to see how it's going to work and how you can utilize it for the tool that you need it for. But yeah, I feel like it's not a matter of what, it's a matter of when, when it'll help solve that problem. That's exciting to me. Even
Mm.
a piece of that phase that takes so many years, if that can condense any part of that over time, that just means more faster, more things out to
Hmm.
market that can save lives and help
Yes,
people
yes,
with
yeah.
disease states. That's so exciting to me. And cheaper, faster,
Well, the
cheaper,
cheaper,
right?
I'm not sure about that.
Yeah,
I'm
well,
not sure about that.
let's-
No, because I mean, you know that there are so many tools. I mean, in the workspace, not in the biotech, in our own spaces,
yeah.
they are free for 1 month. After 1 month, they are 20
Yeah.
euros, and once we all get the license for 20 euros, they ask for 200 because you are going to pay it. I mean, so I think with this it will be the same. It's not going to be cheaper.
Yeah.
That the money will come from other ways, but it's not going to be cheaper.
Yeah, I feel like overall it could be cheaper, but it's not going to be free. It's definitely not going to be free.
No,
I think these tools are definitely going to cost money, but faster. I feel like if it's faster overall, it could be cheaper. If it's
it will be
faster, it'll
cheaper
still
in the
cost.
terms
At
of,
least that's my hope.
yeah, maybe not in the terms of the development, but if you get a specific treatment instead of 20 years in 5 years, you got 15 years more of treatment for people. So
Yeah,
you get less hospitalizations, you get less, you reduce the pathologies. I mean, in other ways, yes, you can spend your money in different ways, but not in the patient, which is good news because we will be healthier.
yeah. Healthier is good. Get it, I'll use that. That's the goal. Well, that's exciting. I'm excited to continue to watch, especially for like the Alzheimer's application, but then just different drug therapies that you can help with as well, and some of these things that haven't been tackled with monoclonal. It sounds like in the past. So on that note, I mean, what are you most excited about upcoming for BioClonal? I mean, and what would be like kind of a dream for you 2-3 years from now, if where you were at?
Well, we are in our first 3 years, and in a startup world, the three first years are quite tough because you are starting
Yeah.
everything and starting all the relationship with your stakeholders and working in the minimal product, minimal viable product. I mean, they are tough. So for the next 3 years, I think we are going to collect what we have now, and for me, it will be very nice if some of the projects that we are starting now, we get the results and we get the candidates for our clients, and we are start to see our little kids around,
Yeah.
because at the end, yeah, these candidates are going to help someone, and it's
Yeah.
very very grateful for a scientist to see how the molecules that you develop in your lab ends in a hospital for
Yeah.
someone.
Yeah. No, that's exciting. You're getting from that point, from all that work, all that time in the lab creation, so much that goes into
Mm.
that. You get to actually see
Mm.
it in patients. So that's
Yeah,
exciting.
it's very.
I'm excited for you all to see that. Yeah, that's a lot of time
Mm.
into that. And then I guess any advice that you have for young people in biotech. I know it's changed, but just from your perspective, if anyone is working on something or seeing something
Mm.
in academia, just any advice that you have for them to make that transition out.
Mm. Yeah, I really like to be in touch with young people in these terms, because I think they are, generally talking, of course, they are quite lost in all this messy world. What I mean with this is, when I study, would you just have to choose between basic science or applied science? I mean, you have chemistry, maths, physics, economics. I mean, there are not so many options. And now, even though, for example, the students that ask me, they are in high school, what should I study? I always answer them, I don't really know, because I don't know what is going to be in the university, in the college when you bring there. For example, bioinformatics. For how long they are studying bioinformatics? 2-3 years, no more. So I don't really know what is going to happen in 3 years. And I can perfectly understand that for them, this is challenging, not to knowing what is going to be in your future, in your very close future. So what I try to tell them is we are going to face a very exciting time, but we have to be very clever in this time. We cannot move with the wave. We have to surf the wave. So I challenge them to be very flexible, to have one objective, that's good, but to be able to run around all the ways that could be in the picture, because we don't really know how we are going to be in 2 years. And I think they are very motivated. They want to change things. I think this next generation want to change things. So I try to challenge them to keep there and not to give up because of this uncertainty we got now.
Yeah, I like that. To surf the wave. You have to be a little more flexible, malleable, which I feel like would have always been good. I wish I would have, maybe been that as well. It would have allowed me to, try more things. But now I feel like, to your point, it's necessary.
Yes.
You
Now
kind
it's
of
mandatory.
that is no question. Like you said, those things moving so fast around you, those are waves, yeah, and you can't just go with them. You can ride them and look for other ones as they come, and yeah, be flexible and malleable. I think that's so key. Yeah, thank you for that so much, and thank you just for your time today and sharing your journey at BioClonal and where you're at now. And yeah, I'm excited to follow your journey and see where you all take this, and excited for you to get these into patients here coming up soon. And just what's the best way to find you all and to keep track of what's going on at your company? Is that just on the website or LinkedIn, or what's the best way
we
to
are
follow?
very active in LinkedIn, and we are quite active also in conferences. We all posted in LinkedIn. So yeah, in any conference or fair that you see us, all is welcome for a nice conversation.
Perfect. Yeah, I noticed that on LinkedIn too, and I'll keep track of that too, because if I attend things in the future, maybe there'll be a time to pop by in person. I've seen a lot going on in Spain specifically too, in biotech.
Yes,
so,
yes.
in Europe,
That's
so
it.
there's
That's it. I think
quite
it's
a
a
bit.
very good moment for Spain, and it's also a very good moment for our specific area, which is Aragon. We got some investments
Yeah.
going on about the Amazon Web Service and so on, and I think it's a very good moment for biotech. We have to take it.
Yeah. Oh, exciting. Thanks so much, Jessica. I appreciate your time today.
Thank you, Christopher.
You've been listening to Stories in Life Sciences, sponsored by MedAccess AI, where we explore people, ideas, and breakthroughs shaping the future of biotech and healthcare innovation. To learn more, visit medaccessai.org and subscribe on YouTube to watch the full conversations and new episodes as they launch. I'm Christopher Wilson. Thanks for joining us. Until next time, keep shaping the future one story at a time.